And we just, in October, we published the paper about reads2map, and the paper tells us about diploid and the comparisons of all the softwares with diploid species. But I already expanded the reads2map features to also cover polyploids. So I will be talking about these comparisons deeply on January with polyploid datasets. So if you want to read about diploids and the parameters of reads2map, filtering and everything, we described it here in the paper. Okay. And so this is reads2map GitHub repository, and you can find here a tutorial on how to set up and how to run it. And it's what I wanted to show you. So as I told you, we go from the read sequences and we do the alignment with BWA, and these alignments go to GATK, FreeBase, Tassum, and Stacks. And from here, you can choose to do the dosage, like the regenotyping with, and dosage calling with Updog, Polyred, and Supermassa. And then from here, we go to the mapping softwares. Okay. And so I split the workflows in two parts. One I call the snip calling. That would be these two first circles here, and the mapping part. That's the read dosage calling, the genotype calling, and the mapping. Okay. After this, with that, these are built in WDL language, workflow language, so it can be automatized and run in different ways. And I have some softwares for doing that, like Cromwell. It's an engine that make all works behind, make everything run in different computational systems, and also making the resources, like optimizing the resources uses. And one thing that's good from all the systems that you don't need to install all the softwares. So you can imagine that each one of the softwares have dependencies and libraries and versions. So I've frozen all of them in Docker containers, and the WDL will call these containers automatically. So you don't need to think about this. You do need to think about how to install Docker or Singularity and Java, but it's only this that you need. And you can run this in cloud environments and also in HPC. And if you go all the way to the end, you have a single output file that's a targz that's input for a Shine app. And I will show you an example of how to run it. But again, if you already tried it and you have questions, let me know. So in the tutorial, I tell about like 3 ways of running reads2map. One is in the cloud, and there's a platform called TerraBio that's made by Broad Institute for WDL workflows that are very intuitive and use Google Cloud behind it. So in case you don't have like computational resources in the university or available, you can have this option. If you do have a computer that's very powerful locally and you have like super user access, you can use a software called Pumbaa. And I need to disclaim here because my brother is the developer of this software. And of course, he's my brother, so I think everything that he does is amazing. But I think it's very useful. And this is what I will be showing in this tutorial. And I will show you what I think is very nice. But for this one, you need to have super user permissions. If you don't have super user permission and you will run your analysis in a high throughput performance computing, you can use another software called Kepper that I thought that was very useful too, because you don't need to concern that much with configurations. You still need to make some changes to be able to set up it. I tried to explain the steps that I needed to do, but it will depend a lot of the system. So how your HPC was configured. Okay, so I included in tutorials some example data sets. I have example for diploid GPS data and also for polyploid, for single end and for pair end. So here I will run the polyploid. Okay, so I will use Pumbaa, and I'm using Docker in my local computer, the lab computer here. And because it's I run just a subset, is the data set that I use as example for running my tests. So I will not need that much resources. So I can run it locally, and if you want to test, you can also run. It will not take that long and will not consume as much resources. So, okay, I will open. So my computer is a Windows computer, and I'm using WSL to have a Linux interface. Okay, I'm in my home directory. So you can see my directories that I have in my home. And I would just create a directory for this tutorial. So here it is. Okay. And I made available 4 samples. 2 are the parents and 2 are the progeny. Again, this is not a real data set. It's just a sample data set, so it's small. So I would just download the samples here, copy and paste, and now I have the data here. This is FASTQ files. Open it will be faster FASTQ format. And I also have this file here that's just the directions for each one of my samples and the name of the sample, each one of my files and the name of the samples. Okay. I will also download the genome, the reference genome. And here it is. It's a FASTA file, okay, with the genome sequence, only part of one of the chromosomes. Oh, I forgot to mention, this is Rose dataset. It's a subset of one of our GPS, and the data was provided by Oscar and Tessa, who did all the lab. So I need to thank them to providing me this data for my examples. Okay. And to run BWA and GATK in the reference genome, I need to create some indexes. I do that with a BWA index, with Picard, and with some tools. But I don't like to install everything in my local computer, so I use a Docker image for that. So you can see the name of the Docker image that I'm using here, and here. So to create this index, I would just run this. And it will be very fast because I already download the images, but if you guys will try this later, maybe the first command that you do, it will download the image and then after use it, so it can take longer. So if I see here now, you will see all the index files for the reference genome. Now I will download the workflows from the repository. If you open here, you can see all the releases. I recently, I think it was yesterday, yes, 13 hours ago, I submitted another version of the workflows, and it's always tagged as the version. And there's also the developed version. That's what I'm using here in this tutorial. But right now, the develop and the main are the same. Okay, so you come back here. And this link is just a direct link to that page. I will just download it pretty quick. Okay. So now I also have the workflow, the WDL workflow. The zip file contains all the dependencies, the tasks, and the other workflows, because I have a main workflow, that's this one, but this workflow call other workflows, and all the others are inside this zip file. And okay, so now I have the main workflow, the dependencies, and a file for the inputs. So this file, I'll open it here. If you see, it will have all possible input files and parameters. And if you want to have a description of what is each one of these, I made also another tutorial specific for that. Oh. Oh. Here. Yes. So this other tutorial have the description of what's each one of the inputs and how you need to define them. Okay. So going back to it, I would just get the inputs that I already defined it here. I just copy from here and replace the template by my inputs. Okay. So here I am using a maximum of 2 cores because, as I told you, it's a data is a subset and my computer doesn't have as much resources. Maximum 2 gigabytes or 2000 megabytes approximately of RAM memory, and the diploidy of my species, in this case is 4. The enzyme we use for the GBS, and if I remove the duplicates, because in whole genome sequence you should remove the duplicates, in GBS you shouldn't, so I put as false. If I will replace the AD, this parameter you understand better, I explained better in the paper, but it's every time that the genotype is missing, I will replace the read counts by missing 2. And in this case, I would not do that. I put as false. And if I will run Tasso, yes. If I will run stacks, yes, again. If I will run JTK, yes. If I will run Freebase, yes. And if I will perform the hard filters in JTK snip calls, yes, I will. And how many chromosomes I'm working with? I'm working with only one. That's the chromosome 4. The chunk size, because reach2map will parallelize the analysis of the samples. And I need to know how much samples you want in each one of the batches that will be parallelized. So you can define it here. The sample file, the file that we just created with each one of the sample files name and the sample name. If I will run mchap, because I'm still working on that, so by now I suggest you to include false, but once I test it, and I will do some announcement and and you can start to put through here. And the genome and all the index file that we just created. You need to be sure that the path for all the files are correct if you didn't make any typo. And I always prefer to put the global path since you're home, okay? And there's another thing that it's a common error, is that a WDL doesn't accept tab characters. So I always use this resource for Emacs that untabify, remove all the tabs, it replaces tabs by space. So if you are using another type of text editor, just make sure that you don't have tabs in this file. Okay, and then once I did it, I have all the files. I have the sample files. So I need to edit here too, because the path, it's not my path, it's the template. So I will come here and I will replace by these ones. Okay, I will save it. Cool. So I have all the files. Now I will run the workflow. So how I will do that? So as I told you, I will use Pumbaa for that. And the first command I will do like Pumbaa local deploy. Actually, I forgot to tell you, but if you want to install Pumbaa, it's very simple. You just run this line. I already have it in my computer, so I don't need to run this line. So I will start from here. Here. Where I was? Here. So I run the Pumbaa local deploy. Here. And it will give me directions on how to establish a MySQL database. And having a MySQL database is a very cool thing to have because it will keep cache of the analysis. And if it happens that you don't have resources enough for the last task of your workflow, you don't need to rerun all the workflow again. It will keep cache for all the jobs that were successful, and we will only rerun the thing that broke. And this is super nice. If you want to change parameters also, if you want to test, for example, just change a parameter in the mapping part, you don't need to run since the snip calling. You can keep the cache from all snip calling, genotype calling, and everything, and just change that parameter at the end. So it's very nice to have. The Pumbaa and the Capper keep cache, so it's nice. I already have my Cromwell database here in my computer, so I can just start it again. And if you check here, Docker, it's here. Nice. So cool. I did this command, so I will ask Pumbaa what to do next. And Pumbaa will tell me, okay, now you open the Cromwell server. So we just copy and paste this line here. So Pumbaa works in Docker, running the local computer, but it also works with cloud, with Google Engine. And actually, my brother configured this mainly for the Google Cloud because it's what he uses. But I asked him if he could adapt it to local, and he did. And that's why I can run only with Docker without sending to computer in the cloud. Okay, so I let it running here, the server, and I will open another terminal here to run Pumbaa. And then I can just ask Pumbaa to submit my workflow. And this is, I just tell Pumbaa submit, and my main workflow, the input file that I just created, and the dependencies, that's the zip file. And to check the status of the workflow, I will just do Pumbaa here. And you can see that I already ran some workflows, but the last one that I submitted, it's still running. And each workflow has an ID. So this submission has this ID. And if I want to see a log of what is happening, I can come to the Cromwell server, and will print with each task that it's making, it was successful or not. And in this case, because I already ran this workflow before, it will not do anything. We just recover the cache, and it will tells me in each one of the tasks that it's getting the cache. So cache hit successfully. So if you see here, the first time that I ran, it took 9 minutes, and now it will just go through the task, so it will take less time. It will be about 6 minutes because it's a lot of tasks, but it's not running anything, it's just going through the tasks. Okay. And so I have 6 minutes for questions if someone have a question. Okay. Or I can also show other things that Pumbaa can do.
Can you put the link in the chat?
Okay. Link for the tutorial?
Yes.
Okay. link. Here to everyone. Okay. So you can see what Pumbaa can do with the help. And one cool thing is that you can go to the metadata of each one of the workflows. So I will get this first one here. And you can see the tasks that the workflows are running. And you can also navigate through the tasks. So you can, okay. And for example, I want to see all the tasks that we are running for GATK. So I can interactively go to the tasks. And this is the hard filtering that I was telling you before. And if I wanted to access the results, this will show me where is it in my computer. So it created, yes, a bunch of directories, but it's important for keeping the cache and keep everything organized. So if I go to this directory, probably I will have access to the images that were created for the hard filtering. Oops. Yep. So it has some questions
in the chat.
Oh, I have questions. Cool. Yes, you can use for parent too. And I have an example. So the question is if we can use for parent. Yes. Someone asked this in an issue in my GitHub, and then we adapted. And I created an example file to make sure that it was working. So yes, you can. And about the duplicates, the GPS, if you remove the duplicates, you lose all your data because all GPS reads are duplicates. So you would not have depth. And so duplicates, I understand that's the sequence that starts and ends in the same position in the genome. So if you remove this and all GPS samples reads are like this, so you will lose all your depth. And WGS, you have like slightly different. They overlap a little bit, but they don't start and end in the same position. And for WGS, these are considered like it can be confounded with errors, the Illumina platform errors when one slot invade another slot, so you get confused about which read it's different or which is similar. So in WGS, we prefer to eliminate that, but because it's not the majority of the data set. But for GPS, it is. So you should not remove for GPS. This is a good idea. So can we have within the reach map a step to merge pair end FASTKey to single end FASTKey? I still didn't think about that, but it's a good idea. Right now you will need to run separately and maybe merge the VCF. But yes, I can think on how to do that all together. Can the input be genotype calls and ask reach to map to imputation to do imputation and construct maps? Yes. So because the steps are separately, if you already have a VCF file, I will show you in the next step. If I have time, because my session's almost ending. But what you can have, if you have the VCF file, you can impute in the next step. That's the empirical maps. just to do the regionotyping and the maps. And if you don't want to do the regionotyping, you just put false in updog, supermass, and polyred. And then reads to map, you use only the calls that you are giving. Okay. If no ploid is known. Yeah, so if no ploid is known, then you probably need to figure out this beforehand. It's actually a good question because we are working on that, and it will be another talk in tools for polyploids. I will not give much spoilers, but we are creating this other software called Kploidy that can be used to estimate ploid. It depends on the data. I don't think it will work with GPS data, but it's working well with like Axiom, and also we recently tested on Dartseek, and it's on the way. But for in this case, for reads to map, you will need to have the ploidy, and you will need to have also the reference genome. And in case you don't have the reference genome, you reads to map, it's a requirement. Can I use software in for AFLP data for exaploid species? I think if it's not sequencing data, you don't need to test all these pipelines. You can go directly to the R package. Like if it's a diploid, you can use one map. If it's polyploid, you can use map poly. You just go directly to R. Let's see if it finished, so I can at least submit it. No, it's still running. And no, it's successful. Yes, it finished. So you can see here, these are the outputs, and it's a little bit messy, but you can see like I output the plots for the filtering. I output the merged band file if you want to use it for another things. Stacks and GATK provide multi-allelic markers. So I separate this in another VCF, and the final VCFs are here in the snip calling VCFs. So, and it lists all the VCFs one after the other. Pumba gives a better interface for that. So I can just get here. Pumba output. I don't remember if it's output or outputs. It's outputs. Here. And then it lists more organized. And you can go and get the VCFs. So these are 4 different VCF because I run 4 different softwares. And so the next step would be I will not run here, but it's available in tutorials, so you can see, you can check later. And if you have questions, you just submit an issue or send me an email. But it's the same steps. Actually, it's just a different workflow and a different input. So here is just the input file is just a VCF. And I needed to change the VCF because for building a linkage map, I need more than 2 individuals. So this time I kept 61 individuals and about 850 markers. And I just upload a different workflow. And of course, the inputs are different, but as you can see here, it's just a VCF. And I can choose to run supermassa, updog, polyred, and if you put false and everything, you just run whatever the genotypes that you input in the VCF. And to run the same thing, just submit it, and Pumba will give you all the interactive ways of seeing what's happening with the run. And the final file, it's a targz file that has the results for the maps. And this file, you can download it, and you can submit to a Shiny app that it's available here. So if you want to install, it's just in GitHub, and you can just run it with run app. The structure is very similar to ViewPoly, if you guys know this other software. And it's just run app, and then you input your files here, and I can get the one that I just ran here. And it will make graphics for all the possibilities. Let me just tick everything here. And this is the number of markers for each one of the pipelines that I ran, and the genetic distance between each one of the markers, like the adjacent distances. So ideally, I want a map that doesn't have like this big distance between markers. So it would, in this case, would be like polyred and updog with a global error of 0.05. And can also see like, so put polyred here. Can also see some overview of the data set that was input in MapPoly. And it explains like the type of markers that I have, the amount of missing data, the dosage distributions, how it was, how many of the markers were redundant, and recombination matrix, recombination fraction matrix for the sequence, and the genetic map. This is just a subset, so it doesn't have much meaning biologically, but it will work the same for a real data set. And also you can access the haplotypes polyred of each one of the individuals. Here, you can choose the individuals and change it. I did this recently, so I will include more tabs because for diploid species you have more options, but I'm still working on the polyploid side. Okay, I passed a little bit of my time. If you have more questions, I will be in the chat or at the end of the session also. Okay, so I think now it's Alejandro time. Hi.
Yes, hello. Hopefully you can hear me well.
Yes, we can. Let's stop sharing. Okay.
Perfect. Okay, so my turn to share. Today I'll be talking about a tool that I have not developed myself. So I'm going to be talking about Polyhaplotyper, which is, let me see, can you see my presentation?
Yes, we can.
Okay, perfect. So indeed, I will be talking about Polyhaplotyper. It's a haplotyping tool that was developed mostly by Roland Forrips, but he couldn't be here today, so I will take over. I'll give a brief introduction on what it can do, and then we can have some Q&A. There is also some links in the Polyploids website with an example data set and an example script, so you can also try it there. But yeah, let's just get started with it. So the idea of Polyhaplotyper is to haplotype blocks of SNPs. So having several SNPs, like what you would get from a SNP array, being able to phase them into multi-allelic markers, basically. It's designed to work mostly for SNPs that are very tightly linked, so very close together, and therefore we expect that no recombinations happen within those very small regions. And again, there's multiple alleles at each haploblock, meaning that it's a way to obtain multi-allelic markers. So in the nomenclature of Polyhaplotyper, the haploblock is the region that you're trying to phase, and the haplotype is each of the possibilities that a haploblock can take. So each unique combination of SNPs. We like to think about this tool like a short-range haplotyper, basically. And yeah, again, multi-allelic markers. So the core idea of Polyhaplotyper, or the novelty that it has compared to other softwares, is that it uses the full SIB family structure to improve the imputation. So most phasing algorithms, they simply use either read information or other types of information to find the phasing, but Polyhaplotyper uses the pedigree information. It works well with full SIB families, but it can also use individuals that are not part of the full SIB family. And in that case, well, it does some optimizations to try to find the minimum number of haplotypes needed to explain all the observed genotypes. You can read more about this whole program in an article that was published in 2022, so last year, by Roland and Giorgio Tumino, who's also participated in this workshop before. So let me tell you a bit the basic principle of polyhaplotyper. So polyhaplotyper starts by looking at a group of SNPs, and let us imagine that we have 3 SNPs. In these 3 SNPs, there's a total of 8 possible combinations of states of these SNPs that make 8 possible haplotypes. But when we look at the genotypes, we don't observe the haplotypes. What we observe are, what we observe are the dosages, but we can imagine that there's 2 individuals that have some number of these haplotypes. So for example, individual 2, that would have 2 times the allele 000, so 0 times the reference allele. And it has 1 time the allele E of this haplotype. So for the first reference allele, so it has one of the chromosomes having 1 time the reference allele, and for the other 2 SNPs, no reference allele, and then the allele F, which is this one. And then we have this other individual that has alleles A and B 1 time each, and allele E 2 times. So in this case, we're looking at tetraploids. I didn't mention this. Of course, polyhaplotyper is also oriented towards polyploids. There's not a lot of haplotyping software for polyploids, so that was one of the reasons we're working on it. But yeah, so these 2 individuals with each 4 haplotypes, in the end, they both end up having the same SNP dosages. So if you look only at the SNP genotype, you will see that for both of them, they have the 2 times SNP1 and 3 times in SNP3 the reference allele. Now, this is the core problem of haplotyping, right? You end up having multiple individuals with similar dosages, but that underlie different haplotypes, and that's what polyhaplotyper is trying to solve. Yeah, this is what we just talked. So the way that polyhaplotyper tries to solve this is by observing for each for each haploblock that you define, it looks at all the possible combinations of haplotypes that would make these dosages. And we need to find which are these correct combinations. The way to solve this problem is to look at the full SNP families. So you look at the segregation across different individuals, and then you end up always finding some individuals that only have very few possibilities. So there's at least 1 haplotype that you know for sure exists in your population, and based on that, you can, let's say, reduce the number of possibilities. Since you know that this haplotype must exist, then it increases the likelihood of some of the haplotype combinations and so on. So you use this information in the offspring to confirm certain haplotypes in the parents, and it iterates this estimation process until it finds a set of most likely haplotypes. For unrelated material, so all of this I was talking is in full sibling populations. For unrelated material, the strategy is a bit similar. So first you feed a full SNP family, you obtain a series of haplotypes that you are almost certain that exist, so you call them confirmed haplotypes, and then you try to see how much of the unrelated material I can explain using these confirmed haplotypes, and it tries to find those solutions that require the least number of new haplotypes to be possible. So this is what we call the most parsimonious solution. So Roland told me a couple of things that I should mention. One of them is that, so you can use polyhaplotyper with unrelated material. It works better if you have at least 1 full SNP population. So that's one thing. He also said that if you have multiple full SNP populations that are related, so that have a parent in relationship, you can use them all together. And the fact that one single parent has two different crosses also improves a lot the quality, or like the amount of haplotypes that you can call, and the certainness that you have in those haplotypes. There's a couple of things to take into account. Before I forget, there was another thing. So sorry, before I mention any of this, another thing was that the time that it takes to haplotype an individual depends on the number of possible combinations that there is for a haplotype, and that really depends on the dosages that are observed. That means that for some haplotypes, even though they might be very nearby each other, so sorry, for some haploblocks, even though two haploblocks might be very nearby each other, one might have hundreds of thousands of possible combinations, and another one might only have 10,000 combinations. Of course, the one with many more combinations will take much longer to estimate. And that means that in the end, the haplotyping can take very long, just because there's a few haploblocks that require a lot of time to compute. So there is a parameter you can set to discard any haploblock that has more than X number of possibilities, and that can reduce the time that you need to haplotype. So the input data that you will need for using polyhaplotyper are SNP dosages. So like, for example, from a microarray, from any other type of SNP marker. And you have to define some haplotypes. So basically give a list of these specific SNPs are the ones that will belong to a haploblock called whatever name you want. And you can define as many as you want. And then lastly, you will need the population structure, so to define who are the parents and who are the individuals. And the main function of the package is this infer haplotypes. And you give here the dosage matrix, the ploidy of your individuals, and the haploblock definitions, parents, and the offspring definition. The main result that you get out of the tool is basically a list. And in the list, there is for each haploblock that you define, there will be a result where it tells you for each individual and each possible haplotype that you have defined. If you have 6 SNPs, I think there are 16 possible haplotypes in this result, for example. So the haplotypes are numbered in binary order, if that makes sense. But in this case, for example, it found that there's only 6 haplotypes that were observed in the population out of the 16 possible. For this individual, it says it has 5 times this haplotype and 1 time this haplotype. For this individual, 3 times this, and 1 and 1 and 1. And you can see that for some individuals, it can find no solution. That happens when there are 2 haplotype combinations. So 2 haplotype combinations that are equally likely or very similarly likely. If there is not one haplotype that is mostly certain, it will just not assign any haplotype to this individual. So in that sense, it's a very cautious program. Yeah, I think that was about it for this. Yeah, actually, that was about it for the presentation. So I wanted to keep it short so we have some time for questions. So yeah, I think actually I will stop sharing unless somebody wants to ask something about a specific slide. But yeah, so that was it. And you can find the links to an R script and some data that you can use to test. That I think is also part of the polyhaplotyper software in this link. So in the Polyploids website, polyhaplotyper can be installed through CRAN. So it just typical install packages polyhaplotyper, and that's it. And yeah, I don't know if there's any questions.
I do have a couple of questions.
Yeah.
This is Oscar.
Hi, Oscar.
What are the limits on number of individuals, number of haplotype blocks, and the size of those blocks with respect to number of SNPs that we can do to look at a survey, for example, of haplotype diversity, if you will?
So let me check so I can tell you the right numbers. So in terms of number of individuals, I don't think there's a specific limit. In terms of number of markers that you can include in each haploblock, it depends on the ploidy. So for tetraploids, you can do up to 8 markers per haploblock. In hexaploids, up to 6 markers per haploblock, and in octoploids, up to 5 markers per haploblock. Yeah, so those are the limits in terms of number of markers. The amount of individuals, I don't think it matters. In the end, it checks individual. Sorry, I think I didn't put it. Yes, there we go, the link.
And how many haplotype blocks can we query?
As many as you have time to wait for the results. So there's no, each haploblock is analyzed independently.
What shouldn't I do with respect to number of haploblocs that I want to query?
Well, I wouldn't do 100 million because you probably will have to wait too long. Honestly, I wouldn't be able to tell you. One thing that for sure you have to check when you set up these haploblocks is that there are no duplicated SNPs in the haploblock. So sometimes there are 2 SNPs that are because they are very close together and they have the same segregation pattern. They will segregate identically in the population. So if you include those in the same haploblock, you're basically having less information in your haploblock. The longer the haploblock, the more information it gives, the harder it is to haplotype. So yeah, that's one thing.
You said that the markers that we choose have to be linked, where there will be maybe no recombination between them. Is there like a threshold there?
So I asked the same question to Roland. He said that in short regions of about 1,000 base pairs, so at 1 kb, that should be good. And in there, depending on your data set, you might have many more than 6 or 8 markers. So his suggestion is within there, you can choose several sets of 6, for example. What you can do then is check how many possible combinations are there of each of these groups that you made. And the ones that have the least number of combinations will be the easiest to haplotype. And therefore, you can essentially find which combination of 6 gives you a haplotype that has the least missing markers and that is the fastest to haplotype. So that's a way to kind of optimize the haplotype search. I see some questions in the chat. Sorry, does that answer all your questions, Oscar?
That's very good. Thank you, Alejandro.
Okay. So a couple more questions. Can we have the number of blocks as optional, then allow the software to estimate? So you have to define the blocks yourself. So I don't think you can allow the software to estimate which blocks. I don't know if that is what you're asking. So indeed, you have to define yourself the blocks. So the software, I don't think there's any specific function that simply gives you suggested blocks to estimate. Can one determine haplotypes in a set of half-sibs first, and then identify the most likely parents in a group of potential candidates? So I think not. The way that the software works is that it uses the segregation in the offspring and the genotypes in the parents to check which haplotypes are the most likely. So you need both the parental and the child information. I don't know if it would be possible, you should ask Roland that honestly, but I don't know if it would be possible to do the full SNP population first. You can use, of course, this parsimonious approach to estimating, let's say, unrelated material, if the material is very related, there should be very few haplotypes, and therefore it might be easy to haplotype them. And afterwards, you could indeed check which of your parents is the most likely. I don't know if it would work very well, though. It's a good question. Maybe it's worth giving it a try. Can you use multiple F1 families? Yes, you can. So in the parameters, you can say who are the parents of who, and you can just set multiple F1 populations all at once. Again, if some parents are shared between these populations, the estimation accuracy improves. Does the heterozygosity level influence the estimation of the haplotypes? So yes, the heterozygosity levels, so more heterozygote individuals are harder to haplotype because there are more possible combinations of haplotypes that would give the same heterozygous dosages. So that is to say, if you have an individual that for 6 markers, it has 0, 0, 0, 0, 0 as the dosage, you know that the only possible haplotypes are all the 0, 0 haplotypes, right? So that's the easiest case. The more heterozygous you go, the more possible combinations can give the same haplotype. So more heterozygous are more difficult to haplotype. Okay. So yeah, I think that if there are any other questions, then I'll be here. I don't know if there's any other questions by microphone, maybe. Well,
Just have a quick one. Are there examples of use of your software in any work, publication, or anything like that?
well, so there is the publication from Roland. I can find the so it's a publication from last year.
But something where they've actually used the software to do some haplotyping per se.
Well, I think they do some simulated and some real data in the article.
Okay.
We haven't used it very extensively, as you can imagine, because it's relatively new software.
Okay.
But let me get the article.
Yeah, why don't you put that on the chat or what have you? Thank you.
Of course, if you have questions about it, you can contact Roland. I would suggest to contact Roland. He will probably answer your questions much more accurately than I can. So, in case you don't know, this is the email for Roland. There you go. So feel free to email him about it. I'm sure he'll be happy to hear if you are interested in his tool. And I will now post the publication link. If I can find it first. And it's somewhere here. Here it is. Yeah. There we go. And here's the publication link. So yeah, and again in the Polyploids webpage, you can find a little example script. It's a bit long, so Roland told me better not try to run it during the workshop because we might have to wait a bit too long. Yeah.
Any other questions for Alejandro? Nobody? Well, thank you very much, Alejandro.
Yes. So I'll see you next week because I will be doing another session.
That's right. We'll see you next week.
Yes.
See you next week.
Until then, well, good evening for me, good afternoon for you, and enjoy the rest of the workshop.
Thank you very much.
Okay, bye-bye.
Okay. Next on board is Jeekin. He's going to talk about map poly. Why don't you go ahead and start up, Jeekin?
Alrighty, share my screen. Here's number 2. Go on. Share. All right. Move all my things in the right places. Okay. All right, so first, I'm gonna put some links. It's a link to a map poly repository. This is where the script is that we're gonna go through. So by now, hopefully, a lot of you, this is I guess the final computational session. So hopefully by now everybody's at least tried Map Poly. So I'm not going to go in and go back and look at other videos of how to run it, but this past year we have some new functionality that helps with filtering and some things like that. So I am going to go over those new functions that maybe or may not be helpful. The tutorial that we're going to follow is that second link I just put in the chat. And to get it, you just press this little download button right here. Download it, and it should open up in your R if you have it. This is what it should look like. And is the text big enough? I think so. Yeah, the text is big enough. So we are going to use a very new version of Map Poly. A lot of these things were just incorporated, or updates to them were incorporated last week or the week before. So you would run this first line to install if you're following along. If not, just watch us go through the tutorial and just view the changes that have been made. That might be more beneficial than trying to follow along. But because I already have it, this is the error that it gives me. And I will load up Map Poly. It's loading. Okay, it's loaded. And there is an example, small example data set that I have put on this link. And what this first thing will do is it will read in the data from this online link. And what I've done in here, we can see here just the name of the file. It's in a subset of data that we have in a population called Bright Eyes by My Girl. It's a rose data set. And I, on purpose, put contaminants in this file. So I have 5 individuals from another population and an unrelated individual cultivar. Right now, if we just print the data DAT, we get some statistics on the data file that we have. Right now, it looks like we have 169 individuals. This marker set only has 2,075 markers, just so that this tutorial runs pretty fast. All of them are actually informative because it's a really subsetted data set. What else can we get from here? We see the number of markers per chromosome, so very few. And we have the different types of markers. We can also see the same data if we plot. We see this. Up here it says no segregation test because I've turned that off. If this is true, what you would see is some segregation tests, and you would see what follows the expected segregation ratios or not. The reason I had it turned off is because in this next thing, this is a new function that Marcelo did. It's called filter individuals. And what it does, it allows you to kind of visually look at your data and filter out individuals that might not fit. So at first, the way that he had it implemented, at least, I guess, a couple months ago, if you ran it, it would just be filter individuals and DAT. And it would use a G matrix that's calculated from what's the software? AGH matrix. And this is an example of it. But you see these guys that don't belong over here, they're kind of close, and they're very, it could be that they belonged in here, but it could not be. Also, if you ran it, you're just using PCAs. Oh, wait, let me see, why is it not? Let me see if I can run this. If you just ran it using PCAs, you kind of really can't tell in the point cloud what belongs and what doesn't. So we're going to come back up here. We're going to allow all the markers just to go through and not filter anything at first. And then using this first method, you see that these groups are, they're a little further out. So with this interactive filtering, you can press Y, and it'll let me draw a box over here, 4 points, or however many points you need. You press finish, and it'll box these individuals. But as you see here, you have 4 points. There's 1 point that's overlapping. There's only 5 individuals. I know beforehand, because I introduced the contaminants, that there's one more contaminant, this SW contaminant. And for some reason or another, using the G matrix method, it didn't pull out that one. So you may need to try PCA. So this was recently Marcelo implemented the PCA method. So let's try this way. Of course, these were already removed. Let's read everything back in again from the very top and try to filter it using the PCA method. And what you see here, there's the 6 individuals that we expect. Let us just circle these guys. You can take however many points you need, finish, draw, and then there you see we have all the individuals that I know weren't supposed to be there. All right. Now, a lot of the steps in MapPoly need the segregation distortion test to have already been run. So kind of the workaround here is we have to read the data in again, and then just remove the individuals that we had identified using these 2 lines. So I'm going to read it in again, filter out the individuals, and don't worry about this output. So this is the proper data set. If we went to print, we no longer have 169, we have 163, so this is right. So we can first kind of look at the distribution of markers. I don't believe this function was here last year, so I'll just run it to show you. What this does is it shows you the distribution, the plotting this, so getting the genomic order of the sequences, putting that into an object and plotting that object gives you a nice, pretty GG plot of the distribution of the markers. So you could kind of run that as kind of QC to see if there's any areas of the genome that you don't have very many markers. And let's say if you know your reference genome, if you have a reference genome and you know that something in the reference genome isn't correct, if there's an inversion. For example, in roses, we have had done multiple maps on the diploid level, on the tetraploid level, and on everything when we align them to the hybrid Saint-Oant reference genome. What we see is there's a small inversion in the middle of chromosome 3. So I know there's an inversion there. And what we're going to do is we are going to isolate just out of this data. We're only going to look at chromosome 3 to make things run faster. This next thing I'm running is I'm running the all pairwise recombinations on chromosome 3. And that should be only a couple seconds. I'll have to make that a matrix. So this is a MapPoly matrix. the number of markers on chromosome 3. You can even plot. I think it's just plot. Let me see. There's a recombination fraction matrix. Looks pretty good. So with this, we can run MDS to look for any of the inversions. So we'll run that real quick. MDS just takes the information from the recombination fraction matrix and orders them. And we can take the output of that, make a map poly sequence. And let's go ahead and run a map using how map poly runs a map. It takes sources of information. You have to give it an input sequence, a sequence of markers. And right now, the sequence of markers that we're giving it is an MDS-derived sequence of markers. We're using the 2-point recombination fraction matrix that we calculated earlier. And these are just kind of some defaults. You don't have to mess with these. So let's just run that real quick. And you'll kind of see it run. I think it'll only take like maybe 60 seconds to run. We should be pretty familiar with all this so far because we've seen this done 2 or 3 times over the years. And we're going to actually be running, I think, 3 or 4 different maps of the same map and just seeing how they compare. So if you have questions while we're running the maps, feel free to put them in the chat or unmute your microphone and just ask. But this shouldn't take too much longer.
Jeekan, you mentioned the presence of this inversion. How do you?
Yes.
How exactly was that determined, and how does the data that you're incorporating now, how is the presence or absence of that inversion across different populations going to affect your potential to map as many markers as possible?
Good question. So you'll see kind of in this tutorial that on the next steps, what we're going to do to kind of alleviate that. But how we found it is all of our diploid maps and all of our tetraploid maps at first were mapped using an MDS method. And MDS method can let you, even though it has very small local inversions, it can let you see large genome scale inversions. So we mapped it using MDS, then we compared the linkage maps with the genomic order, and we found that there were at the same place on every single map there was this. Let me see if I can plot it. First, so we finished the map. You can see the map there. We can plot the map list. So it's about 140 centimorgans. And then we plotted this map. So here you have the genomic position, the physical genomic position on the x-axis. And on the y-axis, you have the linkage map in centimorgans. So there is the small inversion. And on every single map that we did on the diploid, it was probably close 3, 4, around 5 different maps on the diploid level, 2 maps on the tetraploid level. And from other collaborators, I've run their marker sets and 2 or 3 other maps also on the tetraploid level. I'll also have this. So that leads me to believe that the reference genome that was made here has that this is real. If I see it in so many mapping populations, that this is actually real. And if you look at the 2 Old-Blush reference genomes, they actually sequence 2 doubled haploids that come from the same source, and the other reference genome doesn't have this inversion. So there's a comparison paper where they compare the 2 reference genomes, and there's this inversion. So what we can tell from the linkage mapping side is that, well, this particular reference genome may have it wrong just in this small area. So what we'll do now that we know that this guy exists, what we're going to do is we're going to edit the sequence. And this is just another representation, but this is a point and click version. So here's the inversion. We're going to draw a box as carefully as I can. 4 points around these markers. Yep, I captured all those. And I'll come over here and tell it, I can either remove those markers or I can invert them, but I want to invert them. So I'll say invert, enter. And then it'll ask me if I'm happy or if I want to continue. And if I'm happy, I'll say no, and it will invert those. So you see that's gone, and it switched it back to that sequence. So now this edit seek is a object that has edited order, that has what markers were inverted. 24 markers were inverted. So from here, we can make another map poly sequence from the edited sequence. And then we have this new sequence to use. All right. So now we're going to map this again using the edited sequence. And it'll take a little bit, but from the maps, we'll be able to see if there's an improvement or not. So if there's any, let me see, can you see this inversion on the recombination factor matrix? We can probably try to see, but if I go back and hit the button, it'll probably crash my R. So I won't go look at it right now. Can we have the IND to remove based on probability, not based off of genotype names? That would be a Marcelo question. Marcelo might be able to implement something where where it kind of automatically uses a probability-based method to remove. So about 50% through, it's gonna keep going. Keep going. Almost done. Oh, there's another question. The recombination fracture one. All right, I already told them it would probably crash my R. Okay, it's almost done. Let's see.
So Jeekin, as a follow-up to the question regarding the inversion, in a case where you might not know whether the inversion is real or not, either whether it's restricted to the reference or it's something specific to the population that one is working with, would a more appropriate way to handle that be removing those completely and then re-estimating the map?
Good question. So what I have found typically, if you use map poly, and there is an inversion like what we just saw, the reference genome, the reason that inversion is there is because whenever they're assembling the reference genome, there was a same statistical likelihood that it was ordered one way versus the other way. And they just had to pick one way. And it just happened to be inverted differently than what is biologically true. So in my experience, if I just map through that, we're going to map through that just using the regular genome order. It's good enough to pick up QTL, good enough to do any genetic studies. It will map through and keep those markers. So here is linkage group 1 is the MDS map. Linkage group 2 is what we did. We reordered things and we ran a map on the physical location, but this is an apples to apple comparison because this is using kind of MDS and maybe a little more inflated. As you can see from here, MDS has a lot of local inversions, whereas the second map that we ran it's almost like a straight line. You still see this inversion here. We fix the inversion on the linkage map side, so it's still going to show up here in this picture. But the map is shorter, and let me run an example of if we didn't fix it. So let me go ahead and get that started. And Josh, I don't know if I answered your question or not, but this is an example. What I'm running here is if the inversion were there, and I know it's there, I'm just going to map through it and see what happens and compare the maps with it fixed with it and it not fixed. And we'll see.
I'd like to make a comment regarding the question. Is that the germplasm that we use for mapping may come from germplasm where the reference genome that we use is not a direct, I guess, comparator. The assembly that this was compared to was Rosa chinensis. We work with Rosa hybrida, which may have integrations from multiple species, including chinensis or excluding chinensis. Now, the best view or picture that we have for the genetic architecture of our materials really are the genetic maps that are based on the data from segregation of markers. Then we compare that to a reference genome and we may see inversions. It's either because the structure of our genome and the reference genome are not the same, or there are some mistakes along the way. And what we want to do with these physical, I guess, comparisons is try to improve the maps that we have, and we cannot do that across segments where there are clear inversions, either because of our germplasm or because of the reference. And what they're trying to do with these new improvements in map poly is to be able to take advantage of that knowledge and correct for those, to redo the maps that are based again on segregation data nonetheless. So removing that inverted segment would remove a pretty large chunk of DNA. And you may not want to do that if there may be genetic factors that you want to map in that area.
No, of course, Oscar. I'm just wondering, you know, from the biological perspective of trying to determine whether these are true or whether these are potential artifacts of the assembly process, right?
Right. So we've been thinking about that, and one of the things we can do is try to map where those areas of the inversion are and try to see if those are real or unreal, right? We have old blush that is being used to develop the two assemblies that we have for Rosa chinensis. It's not really a true chinensis, but that's what they call it. And we can do some tests to see whether those inversions are real or not, right? So, but that would require additional work that we haven't done yet.
There's a question in the chat that says, if there are some problematic SNPs that we don't have the reference genomic order info, could plots like this help show the location? If so, how? You could use plots like this. You would have to map using an MDS method. And if you do the mapping and you do clustering all with MDS, it should tell you where those, which chromosomes they belong to and where they chromosomes. But you would have to use MDS, and then from there you can kind of manipulate things and assign a putative position or something like that, and then remap it again. And it'd have to be a lot of iterations. All right, so now that we've run those maps, map 1, again, remember, is the MDS map. Map 2 is we fixed the order and ran it on the physical order. Map 3 is if we had just run the map just using its genomic order the way it was. So what we see is the map is slightly shorter with the order fixed. So here's the same plot again, the other plot, and it looks like on 3, so these are reversed again. So this is the MDS, this is with the order fixed. You still see it there because it doesn't actually change the order of the genome. But as you see here, it looks like it just mapped completely through it. You don't see that here. This is when we just map completely through it. Map poly kind of just says, oh, it makes little changes to the dosage calls, and it kind of tries to fix that area. And so you don't see that inversion. So to actually see the inversions, a lot of times you have to use MDS to see the inversion. You won't see it if you just try to map through it, because map poly will either kick out the markers or try to make fixes to the genotypes to make it fit the map. So here, if we look at a summary of the maps, this is the MDS map, 151. And if we map straight through it, it would be 112 and about 104 if we inverted that. So what this tells us is that by inverting that order and then mapping through it, we have a higher quality map, or the mapping algorithm was able to better fit these markers. So as y'all saw earlier, mapping can sometimes take a while. With this small of a data set, it actually maps pretty fast, but other people, that collaborators and even ourselves, if we allow about 10% missing data, we can have anywhere between 1 to 2,000 markers. It can take days to weeks or months for the chain to actually finish, because as you add more markers in MapPoly, and when it's running the hidden Markov chain, it kind of has to, as it's adding markers, check its work backwards. So as you add more markers and you get into the 500-600 range, it takes longer and longer to add another marker. So Marcelo kind of tried to find he found a workaround. So this new function called framework map, there's two functions that work together. He first makes a framework map where it first kind of estimates maps separately for each of the parents and then combines them for markers that are informative for each of the parents separately, and then he combines them. And then another function called update framework map, where he starts adding all the other marker types in. So let me just run it. This is the first section where it is running the framework map. It's running that, and it's relatively quick. And the differences won't be as evident here as it would be on a very large data set. On a very large data set that would take months, now take maybe 24 hours to run. And then this is the second step, updating the framework. So it has made a framework map. It's merged the parental maps, and now it's adding all the other marker types. So as you can see, it's skipping some markers, it's adding markers where it can. And it's working 60%.
So Jeekin, another follow-up question, and maybe this isn't something that you guys have explored yet, but say you have a phased genome assembly with your two independent haplotypes. How are you leveraging that information for the purposes of generating a genetic map? Or are you generating genetic maps independently for each haplotype?
Can you repeat that question again?
So as newer technologies are advancing our ability to generate phased haplotype genomes, how, when you decide, right, to build, say, a new genetic map,
yes,
how are you determining, you know, are you
which one to use?
Which one to use, or are you doing both and then using that for the purposes of comparison, right?
Good question. We haven't run a data set yet with a phased genome, and I know you have a phased genome, so you might be able to do some tests and try to figure out what the best way to go is. And I think yours is also sequencing based markers. So
yes,
we've had some talks, and how I would approach it is I would align your reads to both of the haplotyped genomes, and then kind of combine it together later. And then MapPoly will sort out all the markers that are co-segregated. And it'll keep that in a list so that you know which markers are segregating exactly the same. So you may get more information, you may not, but that would be an interesting test that beforehand, using earlier versions of MapPoly, it probably would be very difficult to test, but now with Framework Map and updating Framework Map, you should be able to do this kind of test and see how efficient it is, or whether or not it's even feasible, or if it's worth the trouble to do the alignments twice, merging the VCFs, and then doing all of that together. We haven't done it ourselves, but it would be interesting to see or to hear from you if you end up trying.
Yeah, I'm kind of at that stage right now where we have our new phased assemblies, and so I'm actively working towards carrying out some of those comparisons. I'm just trying to think about the best approach. First it's chromosome nomenclature, determining which homologs are syntenic with each other, and then doing the active comparisons between the two phased assemblies. But I'm always interested in input.
Yeah. All right, so back to this tutorial. What the update framework map does is it iteratively starts adding markers, and then when it goes through all the markers, it starts again and it tries to add markers again. But you can right now I'm just using the default parameters, but if you don't use the default parameters, you can say, okay, if this increases the map by so many centimorgans, quit adding the markers. Or so what this did, it went through and iteratively tried to add the other marker types to 5 different times. So this last one is your map. So what I'm going to do is I'm going to compare the map lists of everything we've done so far. So this is the MDS map. This is the physical order with it flipped. This is physical order if we mapped completely through it. And this is the new method. We can plot the genome versus the map on all of them. And we can look at the summaries. Oh, actually, I need to change this to 5. We look at the summaries. And what you see on the summaries is they're all about equal, the last 3 that we're looking at. And about the same number of markers. You do see that this do method, you have a little bit more markers that are added at the end. So there's 234 markers that fit, that are added while having relatively the same amount of size. So these 2 sizes are about the same, and I wouldn't say that this is between 4 and between 2. It's not that much difference. All right, so the last thing I want to talk about is a function that I incorporated, that I wrote up. So after we run these maps, what we can do is we can plot what was changed. So earlier I said that hidden Markov chain, after it's running, it makes changes to the input data that we gave it. With information of sequence, with the information of recombination fractional matrix, it makes little changes to the input dosage file that you gave it. So what I want to do is run this plot progeny change on this last map that we ran. And the caveat is that this doesn't work well yet, and I need to make some changes to the MDS or the edited sequences. But this was written a couple months back. So I'll run this. It really quickly runs the error probability. And it gives you a little picture. It gives you a picture of what was changed and what was imputed. So here, all the black is dosages that were not changed. On the x-axis is the number of markers. And then on the y-axis is the individual. So black is the unchanged dosages. Green is what was previously NAs that are now imputed because you know what's on the left and what's on the right, so it can be imputed. And the red is what's changed, and it gives you a percentage. This is the percentage of this entire matrix that was imputed and changed. Now, I also added, if you say output corrected true, what I forgot to do is do this and assign it to an object. Let's say new dosages. I'll run it again, we'll get the same output, but it will give me a data frame of a new MapPoly input data frame that could be used in the future. So this is an imputed data frame right here, new dosages. So this is what you would input to MapPoly. And it's got the corrected dosages. So you could use this for a variety of things. If you've already run a map, you know that this map is what you want, then you could export this and the chain would have to make a lot fewer changes. It actually run through the mapping a lot faster because everything's what it's expecting it to be. With that, I think that's all the changes and edits that I have to share. If anybody has questions, I'm out of time, so 3 minutes over. Feel free to chat in the chat box. And who's next? Do you know who's next?
It's Peter. Yes.
Oh, stop share.
Is that the handover, Jekin? Can you hear me?
Yes, I can hear
We
you.
can hear you and see you.
So I guess that's the handover.
Yes, it is.
Yes,
I
it's
don't
your
know.
turn.
Is someone hosting or apologies?
It's your turn now.
It's yours. Okay, great.
Yes.
Well, my understanding of this session, and I must say I haven't been to these before, is that it's also a venue for people to ask questions. So I suppose I'll leave it open first before I start talking. If there's anyone here who actually uses Polymapper and wants to ask something, I'm here.
Are there any new updates that you've done in the past year that
I will be presenting a few of them in January.
Okay.
In Polymapper, I'm going to be focusing. It's a small update, but it's about mapping in a triploid population.
Okay.
And it's functionality that the package has had for quite a while, but it's something that wasn't really properly done, I think. So I've improved it. So that's going to be part of the January workshop, and a little bit also on some expansions for QTL mapping.
Cool.
Which is maybe a bit ambitious to fit into a single session, but I'll see. I'll speak fast. I just pick up on the question of Joshua. I mean, about phased assemblies. My understanding is that a phase, a reference phase of a genome sequence, it's interesting for an individual, but over a species, I'm not sure how interested I am in a particular phase of a single genotyped individual. If you do mapping with either MapPoly or Polymapper, you will get a phase for that for the parents of that population. So phase information can be figured out genetically pretty quickly. Obviously you need sufficient markers and a sufficient population size to do it well, but I would be thinking that pangenomes are potentially more useful than necessarily tons of phased genomes. But maybe I missed the point of the question, Joshua. Just to link a little bit with what was asked in the previous session.
Well, so I think, in thinking about my question with regards to dealing with biological abnormalities that occur during meiosis or affect meiosis, things like inversions or translocations, etc., with the development of a phased map, or a phased reference, you may have inversions or translocations that are specific to one haplotype over the other.
Yep.
Which could potentially have implications for the purposes of mapping. Although Jekin demonstrated some utilities of method development that actually may aid in dealing with those types of biological abnormalities.
Yeah, but in the situation you're describing, I would expect a serious issue with fertility in that individual.
Yes.
I mean, if there's on a single haplotype or a single homolog, a big inversion and the others not, how's the pairing going to happen in meiosis? So it may lead to issues with fertility.
Yes, of course. I'm just trying to think about ways to overcome biases that result as technical artifacts from the genome assembly process, and ways to exploit mapping populations, and trying to actually decipher whether it's technical artifact versus biological reality.
Yeah, clear.
So it's exactly because we did not see inversions with respect to our linkage maps, but only when we compare our linkage maps to that assembly that we realized that we don't have that inversion in our germplasm, or that assembly is wrong and inverted.
Yeah.
So we would have to do a lot of work to really show whether or not that inversion exists in old blush or not. And we have some ideas how to do that, but we haven't done it. And I know there's quite a bit of heterogeneity in structure among genomes of the same species when you looked at a broad base of germplasm. And one would expect to see cases where there are transpositions, inversions, and other things with respect to one genome to another. But yeah, I agree with Peter. I mean, we would have not been able to map through that area if there was indeed an inversion, because there would be no recombination. It would be inversions are known as recombination inhibitors, right? Where you just basically the whole place just shrinks into a point rather than being able to map through it. So we don't think that inversion really is in our material, or there's no heterozygosity for that inversion in our material, as far as we can tell. Otherwise, we wouldn't be able to map through it, which we have. So I think there's an artifact somewhere along the way. It's probably on the assembly side, and we would have to do more experimentation to prove that.
Yeah, exactly. Yeah, if I'm not sure if it's crossable, if it's all diploid material, Oscar, but I mean, if you crossed old blush with some line which you through your mapping shows an inversion relative to
well, we have material that has old blush in the pedigree, and we don't see that problem. So
yeah,
again, maybe you say, well, the assemblies for old blush are two independent haploids. Maybe they happen to choose one chromosome that has the inversion and the other one that didn't. I mean, that's possible, but we don't see that in the genetic data that we have. So yeah, in all likelihood, it's an assembly issue more than genetic at this point.
I think there's, from my experience with the old blush, there are plenty of assembly issues with both of the old blush, but I shouldn't say that's too loud.
Well, if we look at the dot plots between the two assemblies, you know there are a lot of issues with that
exactly,
right? Yeah.
I should show some slides, I suppose. I have some slides prepared, if that's okay. But I'm very happy to just be here and answer questions as well. I don't need to show PowerPoint slides. If it depends if other people want to ask questions. I'm easy. It's 8:40 in the evening here in the Netherlands, so I'm pretty relaxed.
Yeah, I'm not attempting to hijack the session. I just, these are questions that I cogitate on all the time, because we have these significant issues that arise in our mapping populations, and there's lots of cytogenetic evidence and everything to demonstrate that these are real problems, and I'm just trying to think about how to deal with them as I encounter them.
Yeah.
Some of the things you haven't contemplated, and this goes back to what Chris was talking earlier, is that there's issues with aneuploidy and polyploid populations too. And we're starting to develop the tools so we can detect those and try to do something about it rather than ignore them in some of our linkage mapping efforts. So that's another wrinkle right there, which we all know it's biologically real, and we're just learning how to deal with it.
Hi, Joshua and Oscar. This is Ranjana this end. This is 8:40 in the evening, and really it's too long for us. Yeah, but I still am battling with this yams where we don't know the ploidies, and we have these issues of how to get this, sort these issues when we don't know the ploidies. And I think Agre was asking the same question in the chat. I know that people are still working on it to find some solutions, but do you have
I think we can take this outside of the session, and we'll definitely work with you to try to sort that out. And I'm going to let Peter take his time to discuss what he wants to discuss. And again, we'll discuss this again at the Polyploid workshop per se.
Great.
Hi, Peter.
Okay, great. Well, then in that case, I'll share a couple of slides I had prepared, but I'll go for screen one, share. Okay. And then I suppose I should just now. Do I have to swap my my screens, or can you? Which you're probably seeing my presenter mode. So
Yeah,
so if I do
Okay.
that now, it's probably better.
that's good.
That's
That's
good.
good.
Perfect.
Yeah. Yeah, well, I've just got a couple. Maybe I'll go quickly through them. So for today, I just thought to give you guys a bit of a refresher on Polymapper, and that the package itself, well, as you know, it's developed in a number of different projects. Primarily, hang on, it was primarily developed during a TKI project here in the Netherlands, and then further developed with Dave and Oscar and the SURI project, which is still running. Yeah, and of course, there's the previous workshop presentations. So if you're interested, you can go back to these. Again, this is just a reminder for you that there's actually already a lot of material about how to use the package. I just thought a bit of a refresher on what Polymapper is. It's basically just a set of functions that convert a genotype file with parental information and offspring information into a genetic map. And what is mapping when we're talking about this? So I consider a number of key steps in this data filtering. So cleaning up your data somehow, and G can show some of those steps as well from Map Poly. But we do similar steps in Polymapper. The data is then clustered into chromosomes and linkage groups. And then once we have that information, we do an ordering step using MDS map as well. So we're relying on the same MDS algorithm, which is an extremely fast solver for this optimization problem, finding a good order. And within that process, also we estimate the phase. So we generate phase information in that process as well. So the broad outline for the mapping steps in Polymapper, again, it's just a set of functions. We do a conversion of our marker dosages. We do filtering. We start clustering with the most simple type of marker, which is the simplex, nullplex markers. And then we enrich those clusters with other marker types. We do the ordering, and then we pull out the phase information, and it's done. So it's, in a sense, quite a fast process computationally as well. In terms of what these various steps look like, our rationale behind converting markers to their simplest form is that it's essentially a little bit arbitrary what you call your reference or alternative alleles. And there are, for our purposes, computational advantages to simplifying them where it's possible. What we also like about converted marker scores, at least in the mapping process, is that they give a more meaningful representation of the distribution of important alleles in your material. So, for instance, if I think I have 5 times this SNP times 6 times the copy of the SNP, well, where's my information in this marker type? The information is actually carried on the first homolog carrying the C. So actually, if you convert your dosages, also visually, you get a much more informative picture of where your segregating alleles are across your homologs. We do filtering steps based on missingness as a sort of a proxy for quality. And there are various functions which also allow the user to determine what sort of stringency they want to apply for filtering on missingness across markers and across individuals. We also screen for duplicate individuals because they don't actually add anything, and well, oftentimes, particularly in vegetatively propagated material like rose, mistakes can happen when you're propagating or taking samples. So that's one of the things we filter out. We also do marker binning. Originally, the motivation there was to reduce the size of the data set, particularly with high quality data sets. It's a good idea because a duplicate marker doesn't carry any extra information. It's just a duplicate of another marker. But you can flip it around if you have very high numbers of markers and a more error-prone genotyping technology. You can use duplicates as a sort of a requirement that you need to have a certain number of duplicates in order to trust the quality of that particular marker. So there's 2 ways of looking at binning. And then the calculation of linkage, we remain pairwise linkage oriented in Polymapper. So we only look at pairs, but usually what we find is that if marker density, if there's sufficient numbers of markers, pairwise estimates tend to be quite good, because the most informative ones are from markers that are close to each other. And with high density maps, you're always going to have a fairly nearby marker. So a pairwise estimate is pretty good. Yeah, you could have arguments that the phase estimate is less accurate in those situations, but there are other methods for estimating phase if you really put a lot of importance on the accuracy of your phase information. As regards the output, so the linkage function is the main workhorse of the whole package. Actually, is just one function, and it just produces a table with your two markers, the estimated recombination frequency, shall we say the significance, or at least your assuredness of that estimate, so your LOD score, which gives you a degree of confidence about the estimate, and the phase. Yeah, and in this case, we're visualizing the output of this function in the lower figure there. Following that, we do clustering, and there's two levels of clustering that's performed. One at the level of homologs, so these are these phase, these haplotypes, as you could call them, chromosome long haplotypes. So in the case of rows, for instance, with a base chromosome number of 7, you expect to recover 28 of these clusters in each parent. Once we have these homologs identified, we then need to cluster them into linkage groups. So we have to find, for instance, the 7 chromosomes for rows, the 12 for potato, and so on. And there's various functions within Polymapper and described in the vignette for doing those operations. The clustering of markers is, well, you can imagine it like this. If these are markers, and then what we do is we look at how they cluster together over a range of different thresholds, significance thresholds. So for instance, at a low LOD score, you probably get a lot of clumping together of markers, and you probably don't identify as many clusters as you wish. So because this LOD score is data set specific, I mean, it obviously has to do with the size of your population, what sort of LOD score you should use to cluster, but it depends also on other factors, including genotyping errors and so on, that you might not be aware of. So it's a very data set specific. I can't just say, okay, you should cluster and find your chromosomes at such a LOD score. Nobody can predict that in advance very well. So we took the same philosophy as is used by JoinMap, which looks at how your markers cluster over a range of different LOD scores. And that is quite a good workaround to have a data set specific manner to figure out the structure of your data. In this case, there's an example where if you increase the LOD score up to a high level, well, certain markers will no longer cluster very well. So those are ones that had weaker linkages to the rest. And that can also be a useful step in filtering your data. So getting rid of markers that aren't well clustered. There are various visualizations provided in order to figure out at what sort of a LOD score you should split up your data. And then we use usually we use duplex, nullplex markers to associate those clusters back together to find our chromosomes, because a duplex marker will be linked to markers on multiple of these phased haplotypes. So these chromosome, these homolog clusters. At this stage, there's no order inherent. These look as if they've been ordered, but they're not. They're just bunches of markers. So yeah, the output of the clustering in the case of chrysanthemum would look something like this. So you have now your collection of, in these small little gray circles are your homolog clusters, and the bigger circles correspond then to the chromosomal clusters. So in this case, with 6 in each of the chromosomes. And once we have that, we're more or less done in terms of all the hard work. The clustering tends to be the most difficult step in dealing with data sets, particularly troublesome data sets. The clustering, if you can get something like this, you really deserve a beer or a cup of coffee, because yeah, the main task is done. The rest is then fairly routine. We assign all the other markers based on their linkages to these bigger clusters that we've identified, and then we finish the linkage analysis within each linkage group. Once that's once we have that linkage information, we pass it on to MDS map. So we don't take the approach of, for instance, assuming a genomic order is correct or not. We always de novo re-estimate the order. And that's primarily because it's a genetic mapping software that we've developed, and we want to use the information from the population to figure out the order. And that can always be compared afterwards with a physical map to see how synchronous your population is with the reference order. So MDS map is revolutionary. I often refer to the fact that when I started my PhD in this, it didn't exist, and I was running a join map on multiple computers all over the place, and it was taking me hours and hours to run a single homolog map. MDS map has revolutionized certainly my life, hopefully yours as well. So the output of this process is an integrated map. So it looks something like this in Chrysanthemum. We still need to phase that map. And phasing again means that we go back to our parental information and we figure out how do our markers segregate across these parental homologs. Again, in this case, a visualization from hexaploid species. I won't go into the method behind how we do the phasing. It's pretty simple. I mean, we basically just get the phase information that was estimated during the linkage process, and we phase using linkages primarily to our simplex nulplex markers. It is, I would say, a weak point of the whole process if you don't have sufficient simplex nulplex markers. What I often say, if that's the case, and if you're working in a diploid or a tetraploid, just use polyorigin. It does a fantastic job to rephase. So you can use polymapper to create your map to give an order, and then just use polyorigin. If you're working in a hexaploid, unfortunately, you'll either have to use map poly or just put up with polymapper's phasing, because polyorigin hasn't been extended yet for hexaploids, but it may well be in the future. There are some steps for checking the quality of the map. So again, looking at whether your final order is consistent with the underlying linkage estimations that you've provided it. And generally, unless there's something crazy, it tends to find, at least MDS map tends to find a fairly good order with the consistency between the underlying data. But if this is not looking something like this, well, then you have to think about what's going on and check. Yeah, and that was it. That was a very fast refresher on the Polymapper pipeline. And as I said to Jikin, we will be presenting some updates and doing a more hands-on workshop in January. But I'm happy to take any questions. There's still 5 minutes. So someone says looks good. So that's it's nice to hear. Thanks, Jabam.